Dr. Lindsey Macpherson from the Department of Biology. She is an assistant professor, member of the UTSA Neurosciences Institute and the UTSA Brain Health Consortium. Dr. Macpherson received her bachelor's degree from UC San Diego, her PhD from the Scripps Research Institute, and completed her postdoctoral work at Columbia University Medical Center in New York. Our lab investigates the sense of taste and the molecules, cells, and circuits involved in chemosensation, which is the conveying of information from the tongue to the brain. So she's going to be talking to us about how taste buds work, essentially. I'm sure she'll do a much better job of explaining it and tell me that was all wrong. But
Thanks very much, Matt. I'm not going to have a microphone, so let me know if I'm talking too softly. Usually that's not a problem for me, but let me know if I start to trail off. Okay, so thanks so much for inviting me, Matt, to give this talk today for the Pizza and Research group. It's really nice to be able to talk to a wide variety of students here at UTSA. I teach biochemistry every once in a while, and I teach molecular neuroscience, so if you happen to come into my classroom, be prepared. I'll talk a lot about taste. And today actually is really special for me, especially because it's International Women in Science Day. So it's really great to be here to present my work, to kind of show up for women in science and encourage all of you to continue your research and keep plugging away. We're gonna make parity soon. Okay, so today I'm going to discuss a gut feeling, investigating chemosensory signaling from the tongue and gut to the brain. So this would have been a lot more effective if we actually had pizza, but we'll talk about it and then we'll appreciate it even more later when we get there. So this little girl, she's enjoying her slice of pepperoni pizza. We all know what that tastes like, and we're going to taste it soon. Once you put it in your mouth, what are those kind of feelings you get? You get a taste of saltiness, a little bit of sweetness of the sugar and the crust. You get a little bit of spiciness. You get all of the odors, the olfactory stimuli. So what is flavor? Flavor is a combination of all of those things. It's taste plus smell plus mouthfeel plus a trigeminal sense. So the odors you receive through your nose, it's all the volatile compounds that come up into the air and hit the olfactory epithelium within your nose. Your taste buds can taste only 5 things: sweet, sour, salty, bitter, and umami, which is the sense of proteins and amino acids in your food. And we'll talk in quite a bit of detail in a minute about taste. And you're also receiving information about spiciness in those pepperoni. They contain chili peppers, which activate the pain and temperature sensing neurons that innervate your tongue from your trigeminal system. You're also receiving information about mechanosensory input from the softness, the stickiness, the chewiness of the pizza. And all together, that creates the flavor of the pizza, it really enhances your enjoyment of food. So what would you rather be doing? Would you rather be having a beautiful meeting with friends and family over a lovely meal with lots of different textures and flavors and odors? Or would you rather be like this guy, a tech bro, kind of sitting there eating only Soylent as his only source of nutrients? While you might be receiving the same amount of nutrients that provide everything your body needs, this is a much more enjoyable situation where you're enjoying the flavor of food, you're enjoying the company of your friends, and that's kind of why we enjoy eating so much, because it's a social activity. It's not just a sensory activity, it's all of those things all together. So unfortunately, I study taste, which is kind of boring in the grand scheme of things with only 5 different tastes. But there's a lot of good reasons to study taste. Because it's a relatively simple sensory system, we only have 5 tastants rather than the tens of thousands of odorants that you can experience in the day-to-day with the olfactory information in your nose. But these 5 tastes have really nice innate behavioral responses, and you might know these. You're pretty much innately attracted to things that contain sugars, that contain small amounts of salts, and also that contain proteins and amino acids. You tend to be aversive to things that are highly bitter or highly sour or very, very highly salty. And this taste system is essentially a hardwired circuit from the periphery to the brain. And we can use that information to study how a sensory stimulus can be converted into a behavioral reaction. So these are kind of the canonical tastes: sweet, sour, bitter, salty, and umami. So most of us are familiar with the first four: sweet honey, bitter cabbage, salty salt, and a sour lemon. Umami is the sense of proteins and amino acids within food. And one of the more typical tastes that you might be familiar with that create the umami flavor is Parmesan cheese, or mushrooms, or tomatoes, something that's very kind of high in protein level. So we detect each of these chemicals within our foods with dedicated receptors for those types of chemicals that are expressed on the surface of taste receptor cells. So these receptors have now been worked out. For sweet, we use a pair of G protein coupled receptors, the taste 1R2 plus 1R3 GPCRs form a heterodimer together to allow us to sense sweet tastes. Bitter tastes are detected by the T2R family of GPCRs, which contains about 30 different family members, which together allow us to detect a whole variety of different chemical compounds that are bitter and potentially toxic or damaging to our cells, and things that we should avoid eating. Salty is detected by the ENaC family of ion channels. Sour, actually, was for a very, very long time, the receptor for sour had not been identified, and only in the last couple of years was this gene called autopetrin finally recognized to be the receptor for pH in sour cells. Umami is similar to the sweet taste receptor. It has two GPCRs, T1R1 and T1R3, that together detect amino acids within the proteins of our food. So as I said, taste has a really nice behavioral output. You're usually either attracted to something that's sweet or umami or mildly salty, or you're aversive to something that's very sour or bitter. And this is similar between humans and in mice, so it's a really great system to study. So if you give a baby some orange, it will lick its lips and be very happy. Similarly, if you give a mouse a tastin that contains a lot of sugars, it will readily lick from the spout and want to drink more. Conversely, if you give a little kid a slice of lemon and ask him to suck on it, he'll produce these kind of scrunched up, grimace face look with his eyes kind of squinty and his mouth kind of pulled back in a grimace. And similarly, mice will do the same thing. If you give them a very bitter or a sour substance, they will lick from the spout at first, but then they will kind of pull back their cheeks and squint their eyes and show kind of the same type of aversive reaction to that. So how do we get information about the chemical content of our food that's on our tongue from the tongue to the brain? It requires a neuronal circuit. So within the taste buds on the tip of your tongue, there are a whole collection of taste receptor cells. These taste receptor cells each express the receptors, as I mentioned before, for each of the 5 tastins. And unlike maybe the high school biology textbooks that you might have seen that contain a map of the tongue with specific regions that are sensitive to sweet or bitter or salty, that's completely a fallacy, I'm sorry to say, and hopefully you'll spread this to all your friends, because it's always a question I get. Each taste bud contains taste receptor cells for each of the 5 tastins. So all of your taste buds have the capacity to detect each of the 5 taste qualities, and they're all intermingled within each taste bud around the tongue. There are some slight differences between the front of the tongue and back of the tongue with the actual, relative numbers of taste receptor cells that detect bitter and salty, but overall, you can detect all of these things throughout your tongue. So these taste buds are kind of housed within taste papilla that you can see if you stick out your tongue and look at your tongue very closely under a mirror, those bumps that are present on the surface of your tongue. They house the taste buds. But the taste buds are actually a neuroepithelial cell type, and they don't connect directly to the brain. To get information from the tip of your tongue to your brain to perceive information about what you've just eaten, you need the activity of ganglion neurons, which these ones, the geniculate ganglion, is housed right behind your ear, and they project the peripheral axons out to innervate to the taste buds, each taste receptor cell, and then central projections go and innervate the brainstem. This is kind of the basic circuit that produces those, at least the perceptive part of the reaction to a sweet and or a bitter stimulus creating that attraction and aversion. You get a stimulus here at the top of your tongue. It's received by those taste receptors, expressed by each taste receptor cell. That information is then relayed to the geniculate ganglion neurons, which then relay that information to your brainstem and to higher order circuits or reflex circuits. So one thing that we like to do in the lab is actually image the activity of these geniculate ganglion neurons. We can get a lot of information by looking at a mouse that we've anesthetized and use a surgery to open a window into the geniculate ganglion and look at the activity of those individual neurons to taste sense that we stimulate the tongue with. So pretty much it's a simple assay. We have the mouse asleep, and we perfuse over the tongue sweet, bitter, salty, sour, and umami taste sense. And then we image the activity of those neurons using a calcium indicator known as GCAM. So if we do this, we can image the geniculate ganglion, and we can see that these GCAM responses increase in fluorescence when a specific tastent has hit the tongue. So in this panel, you can see that the neurons will light up when a sweet tastent has hit the surface of the tongue. In this panel, you can see when the bitter cells are activated, salty cells, and sour cells. And these are all within the geniculate ganglion. These are the second order process. So this is after the individual taste receptor cells have bound onto the chemical and then send that signal to the geniculate ganglion. So just as the taste receptor cells within the taste bud are specifically tuned to individual compounds, it looks like we can see that individual geniculate ganglion neurons have a very distinct tuning profile to each of the taste modalities. If we look at these traces of individual neurons responding, we can see how this works out. So each of these traces is an individual neuron, and this graph represents the amount of fluorescence that the GCAM has produced over that sequence of events of the different tastes. So during these windows, we apply sweet, bitter, salty, umami, and sour stimuli, and if you follow the trace of this neuron, it responded very strongly to the sweet stimulus, but pretty much didn't do anything to the other four. This second neuron responded very specifically to bitter. This neuron responded to salty, this one to umami, and this one to sour. And as you can see, if you image lots of these neurons over lots of animals, you can pretty much get the picture that the individual neurons have a very discrete tuning profile, but they will only respond to one of the 5 taste qualities, giving us the idea that taste receptor cells and geniculate neurons are connected in kind of a one-to-one modality-specific mechanism, such that there's this precise labeled line connectivity between the sour responding taste receptor cells and the sour responding geniculate neurons. One problem with this labeled line connectivity model is that we know that taste receptor cells are epithelial cells. They have a very finite lifespan. The average taste receptor cell lives for only about 7 to 10 days within the taste bud. So it's really interesting to think about. You know, you've tasted your sip of coffee this morning, and it's a certain amount of bitter. And maybe 2 weeks later, when all of your taste receptor cells within the taste bud have completely turned over in time, you still have that same feeling of that same amount of bitter in your coffee. So even though these taste receptor cells are continually renewing and replenishing, you have a very consistent perception of taste over time. So this sets up kind of a conundrum. How does this work? Because the taste receptor cells need to connect to the geniculate ganglion neurons to relay information from the taste bud to the brain, we need to rewire the whole system again and again and again. So the taste receptor houses these little taste stem cells at the base of the bud, and they're pretty much the ones that regenerate the taste receptor cells. So as they mature, they kind of come up and become part of the taste bud proper, and then rewire with the neuron axons at the periphery. So one big question then is, how is proper wiring maintained so that you get the same signal over time, even though all of these cells are always constantly renewing? So our idea is that the taste receptor cells themselves must express specific molecules which provide either instructive or permissive signals to establish the correct connectivity between the taste receptor cell and its neuron partner. order to get at what those cues might be, we took advantage of the fact that we can express different colored fluorescent proteins within different subsets of the taste receptor cell types. So we use the bitter gene, the T2R, to drive the expression of a green fluorescent protein in a mouse. And instead, we put a blue fluorescent protein within the sweet taste receptor cell population. We crossed these lines together so that we could get mice where we had bitter and sweet labeled taste receptor cells within the same taste buds. Then we enzymatically digested the taste buds and created individual cells suspensions, which we could then separate into the two colors using fluorescence-activated cell sorter. This is a very common technique used in many biology labs. So we can use the fluorescence color to separate them into two pools. We then prepared RNA sequencing from each of those pools to identify what kinds of transcripts were being created for each of the cell types. So we were thinking that if we could identify potentially a connectivity, a marker, or an axon guidance cue, or a cell adhesion molecule that might give that specificity toward the handshake between the taste receptor cell and the geniculate ganglion neuron population. So we looked at the expression of different genes between the bitter cell population and the sweet cell population. And we thankfully identified some of the control molecules that we were expecting to see. Within the bitters, we found the T2Rs expressed at high levels. Compared to in the sweet cells, we saw the T1R2 and T1R3 GPCRs that create the sweet taste receptor. So we knew we were on the right track. We had good separation between those two populations. Now we're going to look for potential genes that could be used as guidance molecules in the system. And we found two of these molecules, semaphorin 3A within the bitter population and semaphorin 7A within the sweet cell population. So the cool thing about semaphorin is that they are a family of secreted and membrane-bound associated proteins that are widely expressed in the nervous system. And there's been lots of study on semaphorins, especially in neural development, so we had something to go with on this. They're involved in axon guidance. They're critical for neural circuit assembly and synaptic specificity. And so we had these two different versions. One, semaphorin 3A, that's in the bitter cells, and another that's in the sweet cells, giving us kind of like this idea that there could be different guidance cues for each cell type. We then took advantage of the fact that semaphorin 3A has been widely used in the nervous system, and they have genetic models that we can kind of create a mouse where we can delete the semaphorin in specific cell types. So when we did this and we knocked out the semaphorin 3A within the bitter cells, we saw a very strange phenotype within our geniculate ganglion imaging assay. Instead of having the very nicely singly tuned neurons that only respond to a single taste quality, we saw neurons that responded to all of them or a bunch of them at the same time. This is something that we very, very rarely see in a wild-type mouse, so we know something was up. We figured by testing these mice over time that these semaphorin knockout mice lack the ability to correctly connect the bitter labeled line. Instead of just connecting with the bitter neurons to bitter taste receptor cells, the bitter neurons were forming contacts with sour, sweet, salty, along with their normal bitter targets. So this created a very confused wiring system in the mouse. What we wanted to do is to see if that was actually an instructive signal for making these contacts between the bitter ganglion neurons and the taste receptor cells. So what we did was we engineered a mouse where we instead expressed the semaphorin, the bitter semaphorin 3A, in the sweet cell population. And in this case, we were able to then drive the connectivity between the bitter ganglion neurons to the sweet cells, creating mice where we got ganglion neurons that responded to both sweet and bitter stimuli within those ganglion neurons. We tested these mice to see what they would do in a behavioral assay. So we used the bitter compound quinine that's found in tonic water. And in normal control animals, they really hate this bitter substance. And at different concentrations, you can see that they have a very decreased licking rates to 0.3 millimolar and 1 millimolar quinine. The control mice pretty much just avoid it. In contrast, these mice where we had their bitter taste systems rewired to going to the sweet population, they couldn't really tell the difference between water and 0.3 millimolar quinine. They drank it at equivalent levels, and they only started to feel the aversive effects of the bitter at very high levels of the quinine. So these mice would be very happy to go and sip a bitter cup of coffee straight up with no sweetener. So what about sweet? I told you that semaphorin 7A we found was specifically expressed in the sweet cell population. And so we did essentially the same thing. We wanted to make a miswired mouse where we expressed the sema 7A in the bitter cell population, and another one where we expressed the sweet semaphorin in the sour cell population. And we could see that, in effect, we could drive the wiring of these sweet responding neurons to different taste receptors in the periphery, telling you that these semaphorins are really key guidance molecules creating this handshake between the taste receptor cell and the geniculate ganglion neuron. So let me show you this video, and then afterward we'll pause and take a break for pizza.
Freshen up your taste.
Okay, so we had the question of how do the taste receptor cells and the geniculate ganglion neurons get together in a way that they can figure out that, you know, this is a sweet taste labeled line, this is a bitter taste labeled line. And so the nice thing about taste receptor cells themselves is that we have really great genetic control over them. That's how we made this miswired mouse. We could express different proteins under the promoters of the receptors, right, and indicate, okay, we'll express this in a sweet taste receptor cell, we'll express this other thing in a bitter taste receptor cell. The problem then is the geniculate ganglion. We have very little genetic control. We don't know what those different neurons within the geniculate ganglion are doing. So what I was thinking is we could use what we know about the taste system, the connectivity of the taste system, to eventually figure out ways to identify individual populations of geniculate ganglion neurons that are responsible for relaying the taste information about sweet, sour, bitter, and potentially then figure out which ones are attractive versus the ones that are aversive. So kind of following up on the SEMA3A story, we know that SEMA3A is expressed in the bitter taste receptor cells. What's great is that, like I mentioned, there's a lot of work done on SEMA3A, and scientists have figured out that neuropilin-1 is a co-receptor for SEMA3A in a lot of different tissues. So what we thought was potentially neuropilin-1, the expression of neuropilin-1 might mark the population of bitter geniculate ganglion neurons. So to get at whether that hypothesis is correct, we have to do some studies. One first thing we had to do was create a neuropilin-1 Cree mouse where we could express the Cree in the population of geniculate ganglion neurons that typically express neuropilin-1. This Cree line allows us to do a lot of manipulations, and if you go into neuroscience, you'll learn all about this. One thing that you can do is put a fluorescent molecule in there, like YFP, and this is showing the YFP expression in those NRP1 expressing neurons. Another thing you can put in there is channelrhodopsin. Channelrhodopsin is an engineered channel it's an opsin like those found in your retinas, but it's engineered so that you can shine light on it and it will activate an ion channel. It will open an ion channel and pretty much cause the neuron to depolarize, which is essentially a neuronal activation. So what we wanted to do was use the channelrhodopsin as a way to artificially activate the population of geniculate ganglion neurons that are expressing NRP1. Our hypothesis was that if these NRP1 neurons are really involved in bitter taste sensation, activating those neurons should produce aversive behavioral reactions in the mice. So this is essentially what we tried. So here are two videos. I'll play one first and the other second. So first I'll try to start with the control, but it always tends to run this other one first. I don't know why. No, stop. Okay, so for whatever reason, this really got sped up. Mice don't move quite this fast. They move fast, but not quite this fast. So here is the control mouse on the left that is only expressing channelrhodopsin, but it's not activated by, it's not expressed within the NRP neurons. It's kind of like, it's not really doing anything. It's just sitting there, but it's like the genetic control for this mouse. In order to do this experiment, we water deprive the mice overnight. So they're quite thirsty. They're very motivated to go and lick from the spout, but they know it contains some water. So we've trained them a little bit before we do this. So here's the control mouse. So yeah, he's moving real fast. So we have a little light that shines a laser light through the spout. And you can see in some trials we have it on, and other trials we have it off. And this control mouse is very thirsty, and he just drinks willy-nilly. It doesn't matter if the light's on or off. He drinks pretty much full force to get as much water as he can. Okay, that's normal for a mouse. In this NRP Cree channelrhodopsin mouse, we're now expressing channelrhodopsin in the population of neurons that are expressing NRP, which we believe might have something to do with bitter taste signaling. And so when we shine light on this mouse, on the mouse's tongue, he really hates it. He pulls away, and and you can see in the other trials where the light's off, he's still very, very thirsty, and he goes and drinks. I'll show it one more time. As soon as the light hits his tongue, he pulls away. So we're hoping that these types of experiments will kind of shed a light on how the entire wiring and the entire circuitry of the taste system is wired from the tongue to the brain to produce an aversive or an attractive behavior. So now we're going to kind of segue a little bit into taste in the gut. So now that you've eaten some pizza, now we can talk about what happens after you've tasted all that delicious pizza, after you've swallowed it, what happens next? Obviously you don't taste in the gut. You can't really feel what's happening in your stomach as far as oh, I just ate something super sweet. You don't really have a conscious perception of sweet, bitter, umami, etc. in your gut. So, but they are still doing some work. And the interesting thing, and this is a very emerging field, is to look at these. To look at these enteroendocrine cell types, which are very similar to taste receptor cells. Indeed, they actually express taste receptors. So that really makes me intrigued about what is their function within the gut. So these enteroendocrine cells, they line your esophagus, your stomach, your intestines, And they do different things in different places. And very recently, scientists have determined that these enteroendocrine subtypes, some of them can signal directly to the vagal ganglion. So the vagal ganglion is essentially it's another peripheral ganglion similar to the geniculate ganglion, but instead of projecting to the taste buds, the vagal ganglion innervates pretty much all of your viscera, your heart, your lungs, your stomach, your intestines. And it gets a lot of information, including temperature, pain sensitivity, and chemosensory sensitivity, mechanosensitivity. So we have an idea that maybe this connectivity is similar to that in the taste system. So we want to kind of probe what it's doing, what these cells are doing, and what kind of information is being provided to the brain through the activity of these chemosensory epithelial cells within the gut. So in my lab, we're really delving into this project. And one thing we're trying to do is use this technique that I've called TEAMS, tagging evoked activity for mapping synapses. And we can do this both in the taste system and in the gut chemosensory system, hoping to find, like I was talking about, we have a candidate molecule for the bitter taste responsive ganglion neurons, but we don't really have a whole lot else to go for. So another way to find these types of neurons is to link their activity to their molecular identity and a genetic identity would be to do this. So first, our plan is to do GCAM imaging like I showed you before for the taste system. We can also do this for the vagal ganglion. The vagal ganglion is sitting like right in your neck by the carotid artery. So you have to be a little bit careful when you do that surgery. And you can see within the vagal ganglion, if you give the stomach some stretch, if you blow it up with a balloon, you can see these really nice responses within these neurons that are very, very sensitive to mechanosensory stretch of the stomach. So our idea is then to use what we know about the sensitivity of those neurons to tag them with an activity-dependent tagging mechanism, which essentially is like GCount, but instead of just looking at the fluorescence going up and down, we can convert the fluorophore from a green to a red color and pretty much mark those neurons for a long period of time afterward. The good thing about that then is then we can use our tools in the lab to identify what those neurons are expressing, like NRP1 or other receptors that we might be interested in using RNA probes. This would give us a really great idea about the populations of neurons responding to a given stimulus and give us an idea of what those neurons are, what types of genes they express, what types of proteins are they expressing, which is really still a really hot topic in the field. After we can identify kind of the populations of neurons that respond to stimuli, then we want to figure out what the connectivity is. So in that case, we want to look at the actual synaptic contact between the chemosensory cells and the ganglion neurons. So in the taste bud, this would be looking at these contacts right at the taste receptor cell surface. In the gut, we would be looking at these very new, interesting synapses between the gut epithelial endoendocrine cells and the vagal ganglia. And we plan to do this using a tool called GRASP.
Remind me where the vagal ganglia is again.
The vagal ganglion, it's a peripheral ganglion like the geniculate, but it's located in the neck, and it pretty much innervates all of your viscera. So your heart, lungs, stomach, intestines, reproductive organs, pretty much everything in the core of your body is getting innervation from the vagus. And it's responsible for so many things, like regulating digestion, regulating your heart rate a bit, regulating your respiration. So the vagus is really an essential
I've heard the vagus nerve, but I just said vagal ganglia.
Yes, so the vagus nerve is so this is the nerve part of it. The ganglion is the kind of the term for the collection of cell bodies.
Gotcha.
Yeah, so all the neurons, cell bodies are hanging out right up here in your neck, but they have really, really long axons that go and innervate all of your viscera. And interestingly, the central axons go and innervate our favorite part of the brainstem, the NST, which is where the taste neurons also innervate. So there's a lot of crosstalk going on here, which is really, really exciting for me, I think, to figure out if there's some processing that your gut tells you one thing, your taste buds tell you another thing, and you're able to coordinate those two, which is really important, right? If we think about how we evolve as a species and figure out what's a good thing to eat versus what's something that's not good to eat. There's lots of things that are good to eat, right, that we would, like, if you had no parents or whatever, you were out foraging, you would be a lot more careful right, about what you were eating, because there's a lot of bad things out there. I mean, we're kind of privileged now that when you go to a grocery store, you buy anything, you're fairly certain it's not going to kill you. But that wasn't always the case. So this kind of crosstalk between how your gut's feeling. Did I just eat something that made me sick, and what was the flavor of that? And pairing that information so that I'm not going to go eat that thing again because that made me sick, right? So this is kind of an evolutionary process of maintaining us as a species. So yeah, so these are all mechanisms of things that we're going to look at in the lab, and we use a variety of tools. We use the in vivo calcium imaging assay. We use a bunch of genetic tools to isolate those cells, monitor what they're doing, manipulate what they're doing, like with the Channelrhodopsin experiments, and then also look at their connectivity. So I'm just going to talk, super briefly about the GRASP method because it's something that I have developed and really want to use a lot in the future. So I'm going to skip this bit. So this is the idea of GRASP. So at the synaptic contacts, the specific area where one neuron talks to another, there's a synaptic vesicle that fuses with a presynaptic membrane and releases neurotransmitter, which is then picked up by the receptors on the postsynaptic side. We can use this fact of how neuronal communication occurs to our advantage. And one thing we can do is to identify those synapses with a fluorophore, GFP, or a yellow fluorescent protein, or cyan fluorescent protein. So the nice thing about GRASP is that it's based on GFP, the green fluorescent protein. It's split into two parts, and we can put each part on one half on the presynaptic side, the other half on the postsynaptic side, and only when they come very close together, like at the synapse, will they reconstitute their fluorescence, and you'll be able to see that green, the yellow, or the blue color. We can make that activity dependent by putting it inside the vesicles. We can do some fancy things about it. But what I want to use it for first is in the taste system to figure out this connectivity model. Do specific geniculate ganglion neurons connect to only a specific type of taste cell, as would be indicated by our one-to-one connectivity model, or are they more diffuse? Do they contact many different cell types? And nobody's really looked at this yet. So what we would like to do is express the pre- and the postsynaptic GRASP components within the taste system and then identify those sites of contact by the fluorescent color that they give us. So we're creating 3 different mouse lines, one with the bitter expressing GFP construct, one with the sweet cells expressing the yellow fluorescent construct, and then the sour ones expressing the blue cyan colored construct. So then we'd be able to read out at the sites where the geniculate ganglions innervate those taste cells, whether each neuron is specifically innervating only the sweet or the bitter or the sour subtype, or are they all cross-wiring together? That'll really be interesting for the field because it's still an open question. And so eventually I want to apply this to the gut chemosensory system. We know that the gut endoepithelial cells, some of them can form synapses with vagal ganglion neurons, but we don't know which ones and how. So this GRASP method will really be a great tool to identify those kind of synapses, identify which cells are connecting with which other cells, and what kind of information that contact is providing for us for the brain and for digestion. So I'd like to wrap it up just by thanking my lab here at UTSA. My lab is over in the BSc building on the 2nd floor. Come by and talk to me sometime. I'd like to thank my funding sources, UTSA, the NIH, the Brain Research Foundation, and UTSA's Brain Health Consortium. And thank you so much for your attention. It's been a pleasure. Any questions?
When the mice and the laser was being shot, what exactly was it? What exactly was the laser making?
Doing.
Yeah,
Yes.
doing that
Yes.
it didn't like it. And is there a way to actually make a laser that they like?
Yes. So theoretically, yes. So let me rephrase how the Channelrhodopsin works. So channelrhodopsin is kind of like an engineered ion channel. It is activated by this particular laser light, 400 some nanometers. So when you shine that light on a channelrhodopsin, it opens the ion channel, allows the ions to flow through, and essentially activates the neurons in short term. Okay? So what we did was express that channelrhodopsin only within the NRP ganglion neurons. We think those are the bitter ones that sense bitter. So the great thing about the tongue and how the taste bud is organized is that those fibers from NRP neurons, like I said, they're in the geniculate ganglion, that's where their cell bodies are, but they project all the way out to the taste buds. And they get very, very close to the surface of the tongue. Close enough that laser light will penetrate deep enough to actually activate those little fibers that are innervating the taste receptor cells. So instead of activating the taste receptors themselves, we're actually activating the neuron directly. So by shining that light onto the fibers, it will activate the neuron directly at the very tips of those fibers, activate the whole circuitry, and we think that's producing the aversive behavior.
Did you do the same with sweetness?
So we're going to try that. As soon as we have a great driver for the sweet population of geniculate ganglion neurons, we can express the Cre and the channelrhodopsin within the sweet population. In that experiment, we should be able to show that by shining the light on those neurons, that would produce an attractive behavior, and they'll start licking the water at higher, or we'll either give them like a quinine stimulus that they would normally avoid, and when the light's on, they'll drink it like crazy. That's the idea.
Do you think it is possible to actually like replicate flavors like that and be transferable like digitally, so like in the future you can actually like taste something via ions or something like that?
It's tricky. You'd have to make a transgenic human, which, probably that's not the first thing you would target. Yes.
So does the amount of light that you're receiving, let's say you're eating a candlelit dinner, right? Does that affect the strength of taste while you're eating?
Yes and no. So no, it's not like this experiment, right? Because this is a molecule that's not expressed normally in a mouse. It's a molecule that's not expressed normally in a human. We put it there on purpose to do this experiment. So it's completely an artificial setup. But caveat. Yes, like all your environment and all of the different sensory stimuli that you're getting at the same time will affect your ability to discriminate tastes and flavors, but it's more of a central mechanism, right? Because you're getting all of this input at the same time. Kind of when I was talking about, like, what would you rather be, at a nice dinner with your friends enjoying things, or would you rather be sitting drinking Soylent in a closet, right? So all of these things. All of these things really affect your perception of a meal, your enjoyment of a meal. And so things can be heightened, your experience can be heightened by pointing at certain things. So yeah, there's actually quite a bit of research on the combination of all of these multisensory inputs on your sensitivity to different other sensory inputs. So sometimes they enhance, sometimes they diminish your discrimination, your sensitivity.
I have two questions. Question number one is there a commercial application for this to make vegetables taste good? That's my first question. My second question is, how soon till we go to human trials?
So good question. My former lab at Columbia, my PI, he actually formed a company to create artificial tastes. So one, the most obvious is artificial sugars, right? We know saccharin, aspartame, Splenda, it's like sucralose. All of these artificial sugars are great, right? They have 0 calories, they taste super sweet. Anybody who's a fan of Diet Coke is familiar with these things. I hate Diet Coke, I don't know. But, because I think it has a weird aftertaste. Anyway, but yeah, so those are all great things. You kind of get the rush of the feeling of sweetness, but you're not getting the caloric load. There's a problem with this, however, and it goes kind of to the question of the gut-brain signaling, because we have other receptors within our guts that give us an idea of how many calories we've actually eaten, and that satiety signaling is it really important to regulate how much we're eating, right? So if you're giving yourself a whole bunch of sweet taste, you have this expectation that you will be getting a nice heavy caloric meal, right? Because sweet equals calorie, right? Pretty much. Except in the case of Diet Coke, and you're not getting those calories. So you have this disconnect between your tongue and your gut. And we're getting, I mean, this is still, early days in this research, but it really seems to be very important. How you regulate your digestion it starts with kind of some of the taste information on your tongue, getting you ready to take on this load. So yeah, I mean, I think people who have diabetes and stuff, they have to be extremely careful about the glucose and the sugar that they intake. So diet sodas might not be helping you. But yeah, so there's a great interest in making pills that will make the bitter stimuli is really important because lots of medicines are very, very bitter. And so for kids, it's really difficult to get them to take it because it tastes bad, but they need to take it, it's medicine. So creating, like masking flavors on top of it that will either inhibit the activity of those taste receptor cells or enhance the activity of the sweet ones to kind of overcome that bitterness flavor is important. And also salt is another way, right? If you can artificially activate your salt receptor without the salt, then maybe you can, not have all the blood pressure phenotypes. Then salt is also an issue, right? Overly salty. All good questions, and yes, there's lots of money to be made if you can find it. Thanks very much for showing up.